blood group systems. Okay. So our outline, we have ABO blood group, general characteristics, genetics of ABO blood group and H antigen secretor status, and resource blood group system. We will also look at other blood group system, basic red cell serology, immunohistochemistry, pre-transfusion. Okay. So I will also be looking at blood donation, blood bank, organization of blood bank, quality control and assurance, anti-sera preparation, and also look at some lectins. So that is the outline for the course, and our objectives are to, at the end of this course, you will be able to understand the immunological and genetic basis of blood group serology, and you should be able to develop needed skills for donor selection and donor cross match. So that's the outline and our objectives. So we need to define some terms before we go into the blood group proper. So some definition of terms. Antigen, what is an antigen? What is an antibody? It refers to blood group. Okay, as it refers to blood group. So an antigen is a substance that its introduction can give rise to the formation of antibody that reacts specifically with the same antigen, with the same substance, okay? And an antibody is a gamma globulin product of an immune system, also called immunoglobulin, that reacts with antigen against which it is stimulated. And a blood group antigen is a protein or glycoprotein, glycolipids on red blood cells detected by an alloantibody. You should note that the antigens are not limited to red blood cells alone, as we can see later. Blood group systems, they are a group of blood group antigens, okay, that are genetically linked. They are genetically linked. And currently presently we have up to 47 recognized blood group systems containing about 366 red cells antigens as at October 2024 by International Society for Blood Transfusion. Okay, so a bit of history for blood group or the ABO blood group system. Okay, the history of the ABO blood group system dates back to 1900, when Karl Landsteiner is an Austrian physician and is also an immunologist. He made a groundbreaking discovery. Okay, when he mixed some blood samples from different people together, and he observed that some blood samples clumped together while others did not. Okay, so he realized that it was due to the presence of some different antigens on the surface of the red blood cells. And he also identified 3 types of antigen, which he named A, B, and C, C was later renamed O. For the O is a German word for without. So the C was later renamed as O. So his discovery, Landsteiner discovery, was very, very significant because it led to the development of safe blood transfusion. It led to the development of safe blood transfusions. Okay, but before the Landsteiner discovery, okay, blood transfusion used to be very fertile. Okay, Okay, and doctors do not understand why some blood samples were compatible with others and why some were not. So Landsteiner discovery allowed scientists to match blood donors and recipients based on their blood types, which dramatically reduced the risk of adverse reactions. Okay. And for this discovery, he was also awarded the Nobel Prize in Physiology and Medicine in 1930. So subsequently following Landsteiner's discovery, other scientists also made important contributions to our understanding of the ABO blood group system. For example, in 1910, Alfred von de Castillo and Arino Sully discovered the ABO incompatibility reactions. They found that if blood from a person with one blood group is transferred into a person with a different blood group, the antibodies in the recipient plasma will bind to the antigens in the donor's red blood cell and cause them to clump together. This clumping of red blood cell can block blood vessels and lead to serious health problems, including death. So, these scientists were able to discover the, the risk involved, observe the risk involved in blood transfusion, and also try to give some reasons to that risk by evaluating and coming down to describing the blood groups, that there are different blood groups and different antigens with antibodies. So this table shows a summary of the milestone, the key milestone in blood group, ABO blood group discovery, and also other blood group discovery. As you can see here, we have the Lewis, we have the Duffy, we have Kidd, MNS, the P blood, the Diego. So all of them were subsequently also discovered after the initial landmark discovery by Landsteiner. Okay, so we talked about these blood group antigens. Where can we find them in the body? The first slide we were talking about that the blood group antigens are not strictly on red blood cells. They can be found on other body cells. Okay, for example, the ABO blood group is most important blood group system in transfusion and organ transplantation medicine. The ABO blood group antigens can be found on red cells, white cells, platelets, and many circulating proteins. Okay, transfusion incompatibility can cause acute intravascular hemolysis and renal failure and death. So because the red cell antigens can be found also in white cells and platelets and many circulating proteins, it makes it very important during blood transfusion for cross-matching to be done so that to avert or to prevent uh, consequences of any mismatch, incompatible transfusions. So the ABO blood group antigen can also be found on many tissues like the kidney, the heart, the bowel, the pancreas, the lungs. Okay, incompatible transplantation leads to acute humoral rejections. So the ABO blood group system is also important. It plays role not only in blood transfusion, okay, but also in organ transplantations because the antigens can also be found not only in our red blood cells. Okay. So here we have the red blood cell, sorry. Okay, so we have done the definition of terms, discovery, okay. So here we have the red blood cell membrane. This is a very simplified image of the red blood cell membrane. The image is very, very simplified to show some of the key contents of the red cell membrane. Okay, so the red blood cell membrane is a thin, flexible bilayer. Okay, it's a thin, flexible bilayer. Okay, that surrounds and protects the red blood cells. Okay, it is composed of, we have the phospholipids, cholesterol, and proteins. Okay, composed of phospholipids, cholesterol, and proteins. The red blood cell membrane is also responsible for maintaining the shape and integrity of the red blood cell, as well as regulating the transport of molecule into and out of the cell. Red blood cell membrane can perform those functions. The membrane is made up of 3 layers. The first layer consists of glycolipids, glycoproteins, and carbohydrates. This is a simplified image, okay? The second layer is a phospholipid bilayer, and the third layer is a protein cytoskeleton. Okay, the vertical interactions, as you can see in this diagram, okay, mainly involves the glycolipids and the glycoproteins and carbohydrates, and this tends to stabilize the lipid bilayer. Whereas the horizontal interaction, as you can see from this arrow, okay, whereas the horizontal interaction of the cytoskeleton will support the structural integrity of the red blood cell. Okay, you know the red blood cell is a biconcave shape. Okay, the cell has a biconcave shape that is approximately 7.5 microns in diameter. Okay, and it's 2 microns thick. The biconcave shape of the red cells allows it to have a high surface to volume ratio for optimal gaseous exchange, as well as to be able to deform to pass through microcapillaries of 3 microns in diameter. So the biconcave shape of the red blood cells allows it to have a large surface to volume area for gaseous exchange, okay, and also to be able to pass through, change its structure, change its shape, and pass through thin capillaries that are very thin in the microcirculations. So the shape is as a result of the function of the red blood cell. The shape of the red blood cell contributes to the function of the red blood cell in terms of oxygen delivery and being able to pass through very minute capillaries. Then the phospholipid bilayer has an asymmetric phospholipid distribution. Okay, it is semi-permeable and forms a barrier. It forms a barrier between the cytoplasm within the cell from the extracellular environment of the blood. So this layer is also where glycolipids that carry red blood cells antigen and rhesus antigens are embedded. Okay, but as for the membrane proteins, there are transmembrane proteins known as the integral protein, making up the ion transport channels, like the band 3 protein that you can see in this diagram. Okay, these are the proteins. We have the band 3 proteins, the glycophorins, as you can see here. Okay, have the glycophorins. These are proteins in the membranes. Okay, we also have the aquaporins. You can see that here from the diagram. Okay, you can see the aquaporins which transport water. The cytoskeleton are peripheral proteins. which are located on the cytoplasmic surface of the lipid bilayer. Okay, the cytoskeleton is responsible for membrane elasticity, and also for membrane stability. It is anchored to the integral protein via the actin, the spectrin, okay, the protein 4.1 and band 4.2, okay, 4.1, 4.2, the acrin, you have it here, and other proteins that form nodes called the junctional complexes. So this is just a brief description of the red cell membrane. Okay, what we have on the red blood cell membrane is just a brief descriptions, and you can also see the structures that protrudes as the antigens. So the blood group antigens are found on the red blood cell membrane. Primarily, they are anchored to the membrane by a variety of mechanisms. They are anchored to the membrane by a variety of mechanisms, including transmembrane protein and glycophosphatidylinositol anchors. Okay, the transmembrane proteins, as we saw in the earlier diagrams, are proteins that have one or more secretions that pass through the lipid bilayer of the red blood cell membrane. The glycosyl phosphatidylinositol anchors are small molecules that are embedded in the lipid bilayer and have a carbohydrate chain attached to them. The carbohydrate chain of the glycoprotein anchor is covalently linked to the blood group antigen. The type of anchoring mechanism that is used depends on the structure of the blood group antigen. For example, the ABH blood group antigen are all single-pass transmembrane proteins. The rhesus blood group antigen, on the other hand, are multi-pass transmembrane proteins. So that is a summary, just a brief description of the structure of the red blood cell, the membrane. We are zeroing in on the membrane because the reaction we'll be looking at occur at the red blood cell membrane. So we are looking at the constituents, the contents, the proteins, okay, that make up this red blood cell membrane. So you need to know the proteins that make up the red blood cell membrane and why they are there and what they also do. So the significance of the blood group antigens. The blood group antigen is very, very important in the body because they allow the immune system to distinguish between self and non-self. Yes, because when someone with blood group B is transfused with blood group A, there will be a reaction telling you that those antigens, the individual does not have those antigens that is being transfused. So it shows some level of reaction. Reaction meaning it's rejecting that transfusion that is being done. It's not part of itself. So the ABO blood group also plays significant role in self and non-self identification as it concerns blood transfusion. So that if the immune system encounter RBC with different blood group antigen, it will recognize them as foreign and attack them. Okay, so it is very important to match blood groups before transfusion. Okay. 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 There are also different types of red cell surface membrane proteins. The red blood cell also is very, very important. Okay, many of these are polymorphic and have come to attention because they induce clinically significant immune responses when transfused into mismatched recipients. They can cause hemolytic transfusion reactions, like in the first example, or they can cause what we call hemolytic disease of the newborn. Okay, hemolytic disease of the newborn is a condition that can occur when a pregnant woman has a different blood type from that of the baby fetus in the womb. If the woman's antibody crosses the placenta and binds to the fetus red blood cell, the fetus red blood cell can be destroyed. Okay, and this can lead to anemia, jaundice, and even death in the fetus. So the ABO blood group system is very, very important. And currently we have more than 47 blood group systems that are recognized by the International Society for Blood Transfusion. Okay, the clinical significance of blood group in blood transfusion is that individuals who lack a particular blood group antigen may produce antibody reacting with that antigen, which may lead to a transfusion reaction. Okay, the different blood group antigens vary greatly in their clinical significance, with the ABH and Rhesus group being the most important. So upon all the 47 blood group systems that are being recognized, the ABH, which we call the ABO, and the Rhesus blood group are the most important because they exert the most clinical effect. So we'll talk about the ABO gene. They say that the ABO blood group system is controlled by a single gene, and that ABO gene is located on chromosome 9 at position 9q34.2, and it contains 7 exons. Okay, the ABO gene has 3 alleles. It has the A, the B, and the O. The A and B alleles are co-dominant, meaning that both alleles are expressed equally if an individual has 2 copies of them. Okay, the O allele is recessive, meaning that it is only expressed if an individual has 2 copies of it. So the ABO gene encodes a glycosyltransferase enzyme. It encodes this enzyme, and this enzyme adds sugar molecule to proteins and lipids. We'll look at we'll still see more of it. Okay, so the type of glycosyltransferase enzyme that is produced, okay, by the ABO blood group of the system depends on the ABO allele that an individual has. Okay, watch it. A person with the A allele produces an A-glycosyltransferase enzyme, which adds an N-acetylgalactosamine sugar molecule to the H antigen. Persons with the B alleles produces a B-galactotransferase enzyme, which adds a galactose sugar molecule to the H antigen. Persons with the O allele produce a non-functional glycosyltransferase enzyme, which does not add any sugar molecule to the H antigen. Okay. Now, the H antigen is a precursor antigen. It is present on the surface of all red blood cells. It's the precursor antigens on which the glycosyltransferase adds the sugar. Okay, it is synthesized from glucose molecules by a series of enzymatic reactions. So the H antigen is the precursor antigen. The ABO glycosyltransferase enzyme adds the final sugar molecule to the H antigen to produce the A, B, or O antigen, depending on the allele the individual has. Okay. So when you look at this diagram now, this is a diagram explaining what we just talked about. This is the H antigens, the precursor antigens of the ABO blood group, and it contains on red blood cell. Okay, it's present on all the red blood cells. Now we say depending on the blood group of the individuals and also the allele which the individual has. If the person is uh, um, of blood group A, that means it will a person with the A alleles, that is the blood group A, will produce a glycosyltransferase enzyme, which will now add N-acetylglycosaminase sugar molecule to the H precursor, and that is what is happening in this diagram. This is the A, somebody with A allele adding galactotransferase, which is this sugar that it will add. Somebody with B alleles will now add galactose, I think so. We'll add okay, this one is clearer, please. This one will add galactose. This one will add galactose, the B antigens. Then the A antigen will add the N-acetylgalactosamine. N-acetylgalactosamine, while the B allele we add the just galactose to the precursor H gene. Okay. So there are some variations in some things we need to understand about the ABO blood group antigens. Okay, the ABO blood group antigens are designated as A, B, and O. Okay, they are composed of oligosaccharides, that's complex carbohydrate structures attached to the surface of red cells. Their distinctive molecular configuration arise from subtle variations in the arrangement of these carbohydrate chains. Okay, so individuals produce antibodies to the antigens that they lack on their red cells. Individuals produce, you need to understand this very, very well. Individuals produce antibodies to the antigens that they lack on their red cell. Okay, Landsteiner's law states that for whichever ABO antigen is not present on the red cell, the corresponding antibody is found in the plasma. Antibodies to A and B antigen develop in the first few months of life without exposure to the red cell antigen. Okay, as a result of environmental exposure to bacteria, they are referred to as naturally occurring antibodies. The ABO antibodies are usually IgM but may be IgG. So what this slide is trying to emphasize is that individuals will produce antibodies to the antigens that they lack in their red cell. So if an individual is blood group A, that is, it has anti-A on the red cell membrane, that means it's lacking what? It's lacking B antigen. So it will produce what? Antibodies against that antigen that it is lacking. So it will produce what? Anti-B. Individuals produce antibodies to the antigen that they lack. An individual with blood group A is lacking antigen A, has antigen A and is lacking antigen B. So he will produce antibodies, anti-B, that is antibodies to B antigen, which he or she is lacking on the red cell membrane. I hope you understood that very well. Okay, so this is a diagram trying to show us, depict what we have just said. We know that the ABO gene is highly polymorphic gene, that it has different it has different variants of it, meaning that there are many different variations of the gene. Okay, this variation can affect the expression of the ABO blood group antigens, can also lead to the production of abnormal ABO blood group antigens. Okay, here is a table that summarizes the genetics of the ABO blood group system. So these are the alleles present in the red cell antigen of the individuals. Okay, the galactotransferase enzyme is A galactotransferase. So it has the B galactotransferase, non-functional galactotransferase. So the antigen that this galactotransferase will add is the A antigen. So we'll add the B antigen. This one, the H antigen, which is non-functional, it's named O. So then the blood type will come down to A, this one B, this one O. That is no antigen present. So we have what we call the naturally occurring antibodies. They are antibodies that are produced by the immune system of the body without any trigger, any exposure, let's say that, or any exposure to foreign antigens. Okay, they are also known as pre-immune antibodies or regular antibodies. And they are antibodies, they are found in the blood serum of all healthy individuals. And they play an important role in defending the body against infection. For example, the ABO blood group, the ABO antibodies are antibodies that are directed against A and B antigens of the ABO blood group system. Everyone has ABO antibodies in their blood serum, but the type of ABO antibodies that a person has depends on their blood type. Okay, ABO antibodies are produced in response to exposure to ABO antigens from the environment. For example, people with blood type A are exposed to B antigens from food, bacteria, and other environmental sources. This exposure stimulates the immune system to produce anti-B. So that is why they say that the ABO antibodies are naturally occurring antibodies because there was no exposure to any foreign antigen that initiated their production. They are naturally occurring antibodies. Later on, we will look at their characteristics of naturally occurring antibodies. So, how do we inherit? How is the ABO blood group system inherited? What is the pattern for inheritance of the ABO blood group system? Okay, so the inheritance pattern for ABO blood group system, we say that is codominant with incomplete penetrance. It is codominant with incomplete penetrance. What does that mean? Okay, that is that both the alleles of the ABO gene are expressed equally, and that the resulting blood type is a combination of the two alleles. What that means is that if your blood group is AA to say, okay, that means you inherited one from your mother, you inherited one from your father. Later we'll break it down so that you see exactly how it's being inherited. This means that whatever blood group you inherit from your father is being expressed. Okay, is being expressed, the codominance. Okay. However, in some cases, the expression of ABO alleles may be incomplete, resulting in a weaker expression of the blood group antigens. Okay, so the ABO blood group gene has three alleles. We have the A, we have the B, and we have the O alleles. The A and B alleles are codominant, meaning that both alleles are expressed equally if an individual has two copies of them. That is, if you inherit A from your mother, inherit A from your father, you will express both alleles equally. The O alleles is recessive, meaning that it is only expressed if an individual has two copies of it. That will be clear as we go on. Okay. When you look at this table now, it shows the possible blood types and inheritance patterns for the ABO blood group system. Now, look at what we are trying to say earlier. You see parent 1, you see parent 2, and then this is a possible blood type in the baby or the child. So if one of your parents is blood group A and the other one is blood group A, possibilities are that you may be A or AB. Then if one of your parents is A, then the other parent is B, possibilities are that it can be A, you can be AB, and you can be B. This is occurring because of the variations in the inheritance of ABO blood group. When you actually type the genotype, this parent that is A may be AO. This parent may be probably is OO. Okay, this parent is OO, the genotype of the blood group. Okay, so when they combine together, they can either have A or O. Okay, you know, O is recessive. Okay, when somebody is blood group O, it means that he inherited 2 OO. That is 1 O from one parent, 1 O from the other parents. But somebody that is A may be AO or AB. Okay, so the possibility in this combination now, as we can see, is A or O. Then when we have BB, okay, it may be B or it may be AB, because when these parents are characterized, they may be BA. Okay, that's where the A is coming from. So the parent that is BO can always be B and offspring O. They cannot have an A. That is why the possibility that you see a parent that maybe that one parent is A, the other parent is A, they're saying they are AA, and they came down with a child that is AB. Okay, one of them may be AB, one of them may be AO, depending on the variation that occurs in their gene. So that we can be careful sometimes when we are trying to determine paternity. You are supposed to consider the various combination of blood groups that can be possible with some combination of parents' blood group. So that is where it is very important for you as a scientist. It's not when probably a parent is A, the mother is A, the child, the father is A, and you did a blood group of on the child and you got AB and say shouting, how can it be possible? It is possible. I'm telling you, it is possible. There's a lot of variation in ABO blood group, and also the inheritance pattern, codominance sometimes recessive, and then some are not expressed. The expression also affects the blood group. So we have to be aware of these variations in the ABO inheritance pattern and be able to know the possible combination that can happen with certain blood types. That is the message you get here. In some cases, like I wrote here, in some cases, the expression of the ABO alleles may be incomplete. Okay, this can happen when a person has a mutation in the ABO gene, or when they have another gene that interferes with the expression of the ABO gene. Incomplete penetrance of the ABO alleles can result in a weaker expression of the blood group antigen. For example, a person with the A alleles may have a weaker expression of the A antigen, resulting in a blood type that is closer to O than A. So the bottom line of what we're trying to say here is that with a combination of blood group, with a blood type combination, you should be able to decode which type of blood combinations can be possible. And once we're able to say that, you'll be able to not be shocked with some results that can be generated sometimes. Because telling somebody that the mother is B, the father is B, that the child is AB may be weird. But when we explain it on this variations and interference with the ABO blood group gene, and the expression sometimes can be very weak, okay, it's quite possible. So codominant inheritance is a diagram trying to also buttress the point we have made. Okay, this is the gene. Every gene you from either of your parents must show on the upstream when it's operating on this pattern of codominance. Okay, it must be expressed, but not when it shows variations, when there are interference with the expression, okay, of the ABO blood group. That may give rise to other things. So this is an introductory lecture, and we may have to stop here so that next time we'll be able to schedule more